So hello for this second day. So this morning we're going to talk about again Diatom metabarcoding. So there will be let's say the third part. We will talk about reference libraries. We will show you our first proof of concept, and we'll talk about also the impact of biovolume, what it has in terms of impact on the floristic list. Then we'll have a coffee break, and there will be the last part about Diatom metabarcoding after the coffee break, where we'll talk really about the application to monitoring networks, the development of different Diatom indices adapted to metabarcoding. And then Agnès Boucher will talk about an intercalibration exercise that we realized between several laboratories. Then we will have a lunch break, and after this lunch break, it will be the phytoplankton part. And then it will be over for this day. Are you okay with that? Yeah? Okay, great. Up, I put this here. So I hope it's okay with Zoom. Everybody can see on Zoom the screen. Okay, great. So the hornets can start. So here are the schedule I propose you for this first part. First, we'll talk about the reference library that we set up. Then the second part will be the first proofs of concept and the impact of biovolumes. So let's first talk about reference libraries. And so you can recognize here the workflow. We'll talk about the Diatom. So this reference library, which is adapted to Diatom metabarcoding. And here I present you the schedule that I would like to give you. I will make first a presentation of Diatom. Then we'll talk about how we curate the data. I will give you some words about the completeness of this reference library. And the last part will be about the divergence that we can see between classical and integrative taxonomy. Okay, first, let's talk about Diatom and present it. First question, why maintaining a reference library for Diatoms? So as we already told you, there are some reference libraries which exist, but there are mostly some generalist libraries. So you can recognize here this one, PR2, PR2, which means Protist Ribosomal Reference Database. So you have lots of things for 18S. You remember 18S? This second reference library, Silva, which is more generalist but well curated reference library, and Bold, we already talked about that, the barcoding of life database. Then we have specialist libraries. This afternoon, Benjamin will talk about Phytool, which is a library which is dedicated to phytoplankton. This one, PFR2, which is dedicated to planktonic foraminifers. ECREF, Ciliophora, it's for ciliates. DINOREF, it's for Dinophyta. And we could add other reference library which are very dedicated to some particular organism. And for diatoms, so if you want to use 18S, I recommend you to use Silva and PR2, in fact. And we're going to talk about Diatom. And you will see that there are several important things in this reference library. First, there is a really important traceability of data. It's important to know when you have a sequence, to know where does it come from, who produced this sequence, where it has been, where the cells have been isolated, all these kind of things. We also have some photos which are available. And if we don't have any photos, in fact, we put the reference of the publication or the website where you can find the photo. And there is also a demanding curation procedure, which is much more higher than what I mentioned, the libraries I mentioned above. And also what is important is that there are many unpublished data which are made available in Diatom. There is so some reference barcodes from our culture collection, but also from a big project that they had in UK. So they made all this data available and they are integrated in Diatom, and it's helping a lot because here it was a project for rivers. So when you want to work with metabarcoding and rivers and items in rivers, it's very useful. Here you can see the website. I will send you also this website. So this is the address. This is the first page. So you can have some information about the database, how to cite the database. It has been published several times. Yes, here we have a page which describes briefly the content of the library. Here you have the different links where you can download all the data. So, and since there are several versions, so you have all the different versions that you can download. What else? Yes, there are here. It's we propose you a pipeline and a bioinformatic pipeline, which is ready to use, and also an adaptation of a reference library adapted to this barcoding, to this bioinformatic pipeline. There is also a R package that you can use, which is integrated in this pipeline, which has been developed by François Keck, sorry, a former PhD student. Also a page about fundings. It's important to mention that because we have to do that. And yes, the logo. Here there is another page where you can download all the photos. So you can, if there is a sequence and you know we mentioned the culture number, so you can go to this page and all the photos are available for all the cultures. So this is for our culture collection, and here it's for the UK barcoding project. So if you go really to, if you really want to download the database, you have to go on this page. There is a DOI, which is here. Here also, I will send you this if you're interested. And if you scroll down, so you can download in fact, it's a very simple Excel file. I like Excel file. I'm not so very good at making other things, but it's working quite well. And there is another page where we make an adaptation of Diatom for metabarcoding. So here you can find the DOI. This is for Data2 pipeline, and there is another DOI for Mover, which is another bioinformatic pipeline. And the question maybe you're going to ask is, why do we need an adaptation of Diatom? So here you have the full alignment of RBCL. So if you remember, maybe not, the full length for RBCL is 1,005, let's say 500 base pair. So here you have the full length. So maybe you don't see, but you have lines, and all these lines, all these sequences are aligned. This is why you can see these columns, and each color is corresponding to a nucleotide. So you can see that it's quite long, and maybe you remember that to make metabarcoding, in fact, we are working on 263 base pairs because of sequencing limitation. We have to, and also because we have to set some primers. So the length that we are using to identify our items is not 1,500 base pairs, but 263 base pair. So this is this area that we are using here, and not what you have on the left and not what you have on the right. And so when you remove these two parts, some taxa, and mostly some varieties, which were different, so which had different sequences when you use the 1,005 base pairs, when you reduce to 263 base pairs, some of species or varieties becomes equivalent or have identical sequences. So it means that, for instance, I don't know, I'm going to give a stupid example, but Navicula cryptotenella and Navicula cryptoteneloides may become the same may have the same sequence. So in order not to have any conflicts when we make the bioinformatic assignation, we have to adapt the names. So we have to curate again the reference library. Okay. Here, this is the content of Diatom. So this is the last version. 1,500 taxa. You have freshwater, but also marine species. More than 5,000 RBCL sequences. And here on this graph, you can see here on the vertical axis is the number of sequences, and the horizontal axis is the marker because it was not. In fact, there were too few references, and we don't use it. Here in yellow, in yellow, it's a COX-1. We are not using it. And what is interesting for us is really RBCL here in green, and in dark green here too. And here it's the number of taxa that is increasing progressively. And since version 5, in fact, we are not integrating anymore 18S because we have a feeling that RBCL is much more powerful. Here we have a map of the different samples or cultures, the location of the cultures, which were used to produce sequences and which are gathered in Diatom. So you can click on a point and you will see, for instance, here it's the sequence name that is in NCBI. And so it's the barcode is RBCL has not, in this case, probably not a good example. It has no real species name. And so, of course, all these data are coming, some are mostly from here, are coming from us. And here it's coming from different papers which are published in scientific journals. Curation procedure. In fact, the curation procedure, in fact, it addresses a very practical problem of taxonomic assignation. So let's imagine this is your reference database, and it's composed only of 4 species. So it's a stupid example. Of course, we have much more species. And this reference database is not very well curated. You can see here species A. So you have these dots are corresponding to sequences, and the distance between the sequences are in fact the genetic distance. Where here is the field, you make your so you go in this nice river, you take a biofilm, you sequence it, and these two, it's again a stupid example, only 2 environmental sequences. You make your bioinformatic assignation. These environmental sequences, okay, no problem. You can say it's species A, but here you will have a problem. The assignation won't be able to give a species name. It will hesitate. It will be 50-50 between species C and D. So difficult to give a name. So really the curation procedure will be to homogenize each clade, each group of sequence, in order to be able to give a good, easily a species name. So that's basically what we are doing. So yes, the objective of the curation are to homogenize the taxonomical names for a given phylogenetic clade. Why? Because the identification skill of the authors who produce the barcodes, who publish the barcodes, can be different and can evolve from a year to another. So we can have different skills between each other. And also, yes, that's what I wrote here. Taxonomy, you know that taxonomy is changing from a year to another. So we have an heterogeneity between sequences which were published and identified, let's say 10 years ago and now. So we have to deal with that. And also sometimes there are some sequences which are published even in good journal, but the length and the quality of these sequences may be not suitable for correct taxonomic identification. So in some case, it's not possible to take into account these sequences. So the name of the database, sorry, I didn't change. It's not RCs anymore. It's Diatom several years. It has been published, and with this group of people, we are waiting for the photo of Daniela. I'm pretty sure that we will be able to add your photo in a few months, thanks to the BiolaWeb project. And so this is the procedure that you can see in the paper. I'm not going to detail you this, especially for those who are in the back of the room. It's too small, but I'm going to give you an example of curation, which was done by Jonas Zimmermann, who's working in Berlin in the Botanical Garden. It's a curation which was carried out on Planotidium. So this is a part of a phylogeny. So it's a tree. So sequences which are near in the tree have similar sequences. The distance between the sequences are near. So each line here is a sequence, and we replaced, of course, the sequence by the name of the species. And here, in fact, it's the code of the sequence. And you can see here in this nice tree, there is a nice bootstrap, so but we have different names. We have a majority of Planotidium capucium. We have some Planotidium frequentimum who are in the neighborhood, and Planotidium sp. So what do we have to do? In fact, we have to go back to the publication, have a look at the photos, and it was possible for this Planotidium capucium. All this Planotidium capucium, there is a publication, and there are some photos which are available in this website. So we can check if the identifications are correct. Here, Planotidium frequentissimum, it's coming from our culture collection. So again, here it's possible to check the photos which are on the website. Here, we are lucky too, there is a publication, so we can check the photos. And this one, it's from the team of Edouard Theriault in the United States. And he deposited a photo of his culture in this website. So we were able to, Jonas Zimmermann was able to change the name from Planotidium frequentissimum in Planotidium capucium and so on. So now we have an homogeneity of the species names in this clade. However, I told you that year after year, we have changes of taxonomy. Planotidium capucium, I think it was 1 year after we did this job in 2018, change name, or someone say, okay, there is a synonymy between Planotidium capucium and the Planotidium victorii. So in the version just after, we had to change all these names in Planotidium victorii. Yeah. So that's basically the work we are doing to manage to have a homogeneous taxonomy for each clade in Diatom. So it's really step by step, but to reach to have something correct. But there are still some mistakes for sure. And if there are some people who are ready to help, please, no problem, come. There is a nice group, and standardization about these Diatom libraries. In fact, quite early between the colleagues who were working with Diatom and metabarcoding, there was a general agreement that it was really important to have a metadata traceability. First thing, and second thing, that this reference library remains open access, so accessible and freely accessible. Also, what we were agreeing quite quickly is that the vouchers of the specimen which enable to produce the sequence must be deposited in collections which are open to everybody. So museum, botanical garden, culture collection, in order not to limit the access to this material and to be able to check if things are correct. So it can be slides, strains, dry or frozen material. What kind of metadata should we keep in the reference library? There is a problem? Yeah. I can give you mine. Yeah.
Can you explain the voucher? What does it mean?
Voucher, in fact, it's a material who the voucher is the material who is deposited in the museum, and where, for instance, you have a new species which was described, and so you have a voucher of, how do you say, the material which is deposited in the museum, the specimen. Yeah, yeah. Is it correct? I know there are some people working in museum much more than me.
We also have collection. Yeah.
In collection?
Yeah.
Okay. Yes, exactly.
Species for science, then we have collection.
When you produce molecules, important to say how you produced it. What kind of sequencing technology? Is it a single sequencing? Is it Illumina or whatever? Of course, the marker. Is it 18S? Is it RBCL? It's important to say it. It's important also to tell what kind of primers you used, the extraction method, the PCR protocol, the dates, the names of the persons who produced that. Also, if it's coming from a culture, the culture details like the isolation date, the name of the isolator, maybe the culture medium, the strain identifier. And when you put a name for a culture or a sequence, of course, give the species name or let's say a taxonomic name. But it's good also to know the name of the person who did the identification and the literature he used, what kind of book he used to give this taxonomic name. It can help also to see if we can have some differences between us. And also it's important to add photos. So we were agreeing about all these criteria, and we started standardization in 2012. We went to the SENS, of a European Committee for Standardization. And 6 years after, so we had this technical report who had been accepted and had been translated in fact in French, but in all other European countries' language. And here you can see the names of the people who helped in the standardization procedure, especially in writing this technical report. We also, so it was 2 years ago already, we also had a work for standardization, not only for diatoms, but for all aquatic organisms. In fact, we were a nice group of people working on fish, on fungi, on cyanobacteria, all kind of aquatic organism you can imagine. And the idea was to set the different metadata which were important to add in the reference libraries. And we put all this information in this paper that you can find in MBMG. And this paper, it was really, I would say, an output from Diana Quanet, so this European cost action. So Agnès Boucher and Florian Lezer were co-leader of this nice action, and there was a nice group of how many people? Maybe 400, yeah, 400 people. So it was really an extremely enthusiastic group with lots of exchanges. And this is one of the example of output that we had in this nice cost action. Do you have questions about these parts, things which are a little bit fuzzy or no? It's clear. It's clear for you, but maybe there are other people it's not clear. No? It's okay. So let's continue. Completeness of Diat barcode. So you see the different questions. How can we complete the library? And I will show you how is it complete in France, in the rivers of France, but also in other habitats. How can we complete the library? Maybe I should have started from here. The classical way is to make some cell isolation. So you take your biofilm, it's full of different species, some mushrooms, some everything, some bacteria, and you have to isolate one cell of Diatom. So you can use directly by pipetting under, usually it's an inverted microscope. It's long, it's quite tedious because there are many species that they don't like being under culture. So sometimes it's not using, but this is the classical way. And after that, you make a Sanger sequencing when you have enough biomass. There is another solution. It's to make single cell PCR. So you take one cell and you try to identify it with on living material, and directly you sequence it. You make a PCR on the single cell and you sequence it. It's working. So there is a paper, a nice paper from Hamilton. So they reached to do that for several. I remember it was some Storonis, if I remember well. So it's okay for big species, but when we are talking about small navicula, small things, it's much more difficult. Another solution is to make some cloning sequencing of natural sample. I would say that it's a little bit the ancestor of high throughput sequencing. The problem it's quite long. The good thing is that it's producing long sequences of good qualities because it's basically some Sanger sequencing, but it's quite expensive and can be done for samples with only can be done for samples with low diversities. We propose to use, in fact, high throughput data to complete the reference library, and I'm going to explain you how we can do that. The idea is, in fact, to select samples with target species which have never been sequenced. I give you here an example, still on Mayotte. This is Kalman here on this photo. It's a little bit far. And in this river, there was one species, this one. It's an Alamphora. You can see it on the scanning microscope. It was dominant in the sample. And when we make the high throughput sequencing, it was evident because there was one sequence which was dominant and which was not determined. So we could easily make the hypothesis that this sequence should correspond to Alamphora ghanensis here. So what we did, in fact, so yes, what you need is to join the microscopy to the DNA, and you can make a phylogenetic checkout. So you can insert this this sequence, this environmental sequence in the phylogeny. And here it's okay because it's really in the group of Alamphora. uncertainty associate this sequence to this nice anaphora organism. So there is a publication associated to this methodology. And basically, to complete quickly the Diatom reference library, these pieces which were not sequenced, we didn't have the reference in the library. So by selecting good samples with these dominant species not sequenced in high abundance, and we could make a correspondence with environmental sequence. So this is an example here. It's Acanthidium delmontii. So now we have the sequence of Acanthidium delmontii. And we did it for several versions. And here you have different regions of France. So this is mainland France here in blue. You can see here, so with the different versions here, the percentage of reads here, which are assigned to species level. So you can see here, for instance, V7, 83% of the reads were assigned to species level in France, in mainland France. It was not the same stories in tropical areas. Here you have, if I remember, yes, here in red, you have French Guiana. French Guiana, we are really in the Amazonian forest, and it's full of diversity that is very complicated. But progressively, at least for some areas, here it's Île de la Réunion in the Indian Ocean, we were able to complete quite well the reference library. Here also you can see it coming from Zlatko Levkov laboratory. She was able to complete the reference library for these two islands, Martinique and Guadeloupe, which are in the West Indies. So we reach 80. But yes, you can see here, French Guiana. In fact, even for microscopy, we don't know the species. So we don't know the species in morphology. We don't reach to make any correspondence. It's really too complicated, and we don't have. It's possible to use this methodology when the species are well known in the region, but when we don't have any certainties, even in microscopy, it's not possible. Completeness. Again, so until now, we have a library complete. And so still in my yacht, there are sea turtles. It's a very nice place in the south of the island, and there are sea turtles everywhere. In fact, we asked the permission to have some biofilm from the shells of the turtles. We made some high throughput sequencing. There is a nice paper of Cinziana Rivera in +1. And we made the taxonomic assignation with DNA metabarcoding and microscopy, because there are some diatoms here and it's quite nice diversity. Here you can see that with microscopy, we reached to identify 63% of taxa to species level. And here it's a genus, I think it's, yes, it's species or which are probably new. And here 12% to genus level and 7% we really didn't have any idea, because it was very small, and we needed we would need really some in-depth scanning microscopy. We started, but it was really too important taxonomical works. But you can see that 63% of the reads we could put a species name. With DNA metabarcoding using Diat barcode, only 10%. So you can see here there is really a gap for some habitats and in these very particular environments. Okay, do you have questions about Diat barcode? No questions? Crystal clear? So if not, I will continue. Yeah, Alexandra, yeah, please. Sorry. Yeah. Mhm.
63% is okay. Yes. You determine 63% using microscopy and only 10 by my
So here.
metabarcoding. Yes, but how can you not use those samples to add them to your database? So later you can. Did you add those codes now in the library? Or
No,
how-
no, it was really a lot of work. It would have been. In fact, there were many. Yes, there were too many unidentified sequences to have. Well, it's possible to complete the reference library when, let's say, 80% of the reads are identified, and then you have only 20% which remains. It's okay to complete with microscopy. Or it's possible when you have a high abundance of one species which is not identified. So here, yes, you can make the link, but here there are too many uncertainties. Like in French Guiana, yes, the taxonomy is not well known with microscopy. We don't understand the morphological boundaries of taxa because you have also this cell size reduction. And yes, we don't have enough references in microscopy, in morphology, and the DNA tells you again a different story because it's also the way people count the diatoms in French Guiana under microscope. It's difficult. They put some boundaries which are sometimes, I think, probably the same. So maybe they put some boundaries, for instance, for two species which are probably the same species, but with a different cell size reduction moment. I mean, we could realize that, but it was really too difficult to see there was a too important gap between the two kind of data. And here with the sea turtles, it was also a big gap. So it's not. I think it's too long to be able to to feed the reference library with this methodology I showed you just before. I don't know if I understand. I answer correctly.
Yes, thank you.
Yeah. Yeah, Sergeant.
Get their barcodes. From the Zoom, the question, do you focus on indicator species to get their barcodes?
We focus on really, for mainland France, we focus really on the dominant species which are not barcoded. It's the easiest way to complete the reference library. When you have a lot of data, okay, you can give a name to these unknown sequences with more confidence because you can also make some correlation between DNA and microscopic data. You can make several phylogenetic assignations in the tree. I mean, so we are much more confident with dominant species than in rare species. Yeah. Rare events is always complicated. Yeah. Okay, thank you. Other questions? No? So, and then we already started to talk a little bit about this divergence between, let's say, classical and molecular and integrative taxonomy, just with these questions. Here, yes, I would like to talk about that. Some divergences that we can have between, let's say, classical microscopy and the way that we can define the species boundaries with more criteria. I would like to talk about that at specific level and deeper nodes. So when I'm talking about classical taxonomy, it's when you define species boundaries based on a single criteria. And for diatoms, usually we use fuscule morphology. And based on this fuscule morphology, you say, okay, we have one species and here are the boundaries. And the problem is that we have this phenotypic plasticity. I was talking about this problem that we have in French Guiana. It's quite complicated. So you can have problems to make the delimitation of species with classical taxonomy. The idea of integrative taxonomy, so it was introduced by Diatom in 2005. The idea is to make this species delimitation based on several criteria. So you can use morphology, you can use DNA, but you can also use ecological information. You can use also, for instance, interbreeding experiments, so to see if species, if two individuals are able to breed and give some descendants are able to survive. So you can use all these criteria. And I would like to give you a few examples of using integrative taxonomy for diatoms. The first example I give you is a very nice paper of the German colleagues of the Botanical Garden of Berlin, and especially of Regine Jeanne, and there is also Jonas Zimmermann. And this is an example of Planotidium. The first thing they did, so they had many cultures of Planotidiums. You can see here the different lines are each time it's a different culture. So they have really a nice person making many cultures of his which are neighbor here, for instance, these two. It means that their morphology is similar. And they were able to define a certain number of groups which are well supported. So we should have a bootstrap value here. So you have different clades here. They did the same thing with DNA. So each culture, they sequence each culture. So again, here you have all the cultures, but instead of morphological criteria here, it's the molecular criteria. They look at the distances between each sequence, and they produce again a second tree. So here it's based on RBCL and 18S. So two different markers were concatenated to produce this tree. And you can find again a certain number of clades. And they were able to combine these two criteria, morphology and molecular criteria, to delimit a certain number of species. So you can see here, this is one species, this is a second one, a third one, and so on. And for people who are who are using classical books of identifications, so the Kramer-Lange-Berthelot books, and where we are making classical identification for routine monitoring. When you have these species, it's quite frightening here, but making the difference between this shape and maybe this shape, it's really difficult. But they found some tiny different morphological differences and some molecular differences. And it's really the combination of these two criteria which were able to define with robustness these species. So just to make you understand that, yes, there is more and more a gap which is starting to increase between the classical books that we are using and this new kind of delimitation based on integrative taxonomy. I would like to give you a second example. So here it's also going to speak really the diatom is it's Gonphonema palum, and this is a paper of Lenaï Kermarek. She did the same thing. We had a certain number of 39 cultures. She sequenced 18S, ITS, RBCL. This a little bit doesn't matter, but you have groups. She defined clades. First thing, she did the same with morphology. So we had this culture, and she made some morphometrics. So she put some points here at the different of this rise multivariate analysis, and she was able again to define some clades. And the best combination of a morphological criteria and the DNA data were these points here. So this area of a frustule. When you open the classical books, Elena, tell me if I'm wrong. When you open the classical books to identify Gonphonema parvulum, we are not looking at that. Daniela, tell me if I'm wrong, but usually we are counting the numbers of stria. We are looking at the width, the length, what else? The poles, we are looking at that. This, there is a divergence between what these last years and emerged from this combination of DNA and morphological data. And the colleagues of Berlin, in fact, paper species based on the same kind of data, and which these species are also very difficult to identify purely on morphology. Okay, do you understand what I mean? And do you see what also what can be the impact on the floristic list that we can produce with metabarcoding and with microscopy? Okay, so you understand. Okay. But we also, well, it's less important for metabarcoding, but we also have a divergence between the classical taxonomy that we learned in the book, for instance, of Randman and Crawford, and the more recent results that we have with molecular data. Yesterday I presented you this traditional classification based on these 4 classes. And there is a nice paper, a really nice paper of Edouard Thériault and his team, so the American colleagues. What they did, so they had more than 200 cultures, and for these 200 cultures, they had 6 genes, so 1 nuclear, so I think it's 18S, 4 chloroplastic, and 1 mitochondrial, so COX-1. And they were able to reconstruct with really a lot of certainty the deep nodes of diatom phylogeny. It's really because they had this length, these numerous genes put side by side, that they were able to make with confidence this to reconstruct these deep nodes. And you can see here what is very interesting. So is that all the groups are paraphyletic. And this is a problem for taxonomy, because taxonomy just a reminder of what is a paraphyletic group. This is a paraphyletic group, this is a monophyletic group. And as you may know, systematic taxonomy only accept this kind of groups. But for diatoms, we only have the large majority of the groups, in fact, mostly all, are paraphyletic. So it's a problem for taxonomy. And so in the paper of Adel, 2019, this one, so I already showed you this tree. David Mann took into account the results of the colleagues, American colleagues, Edouard Thériault and team, to produce a new taxonomy for diatoms. And this new taxonomy is integrated in Diatom. So it's integrated, but I also kept the classical taxonomy too, not to be completely lost. Okay, that's all for reference libraries and what you can see between this diversion between. I don't know if I'm too late. Maybe I'm taking too much time. Oh, until what time do you have? 8 minutes? Oof, sorry. So the first proof of concept. Sorry, I was too long. The first proof of concept So we carried out with Lenaik Kermarek on 4 samples that you can see here. And we wanted to have a large diversity of samples. We made a 454, sorry. Okay. Okay. No, but so you told me until what time I. Okay, so 1/4 is okay. 15 minutes is okay. Okay, so let's go. Okay. Okay, no, but in fact, it's not so. It's interesting. Here is just to present you our first proof of concept that was carried out. It in the paper of Lenaik almost 10 years ago. And what you can see here is so you have 3 points on this NMDS. You have microscopy, RBCL, and 18S. And each time you can see that there is a good congruence for each sample. So here is one sample, a second sample, and so on. So it was our first proof of concept. The second one, it was carried out on Mayotte Island. So I told you we presented this first proof of concept of Lenaik to the water managers. The water managers, they say, okay, we are interested, go in Mayotte and try to do this again, but on a larger number of samples. So we went there. So it's here between Madagascar and Mozambique. So this is the island. It's very nice. Here you can see so the Kalman is here bathing near the sea turtles. What you can see here. But we were more interested in rivers. So there are nice rivers in this island when you go in the mountains, but you also have very polluted areas because in fact, there are lots of immigration and in fact, how do you say the bidonvilles? How do you say? You know, when yes, you have houses which are constructed with slums. Okay, and so there are many things like that, and people are putting all their rubbish in the water, especially in the downstream part of the rivers. And also there are many ladies, almost all the ladies are washing their clothes in the water because it's their classical way to use rivers. But the problem is that rivers are quite small and the population is very important. I think it's 500,000 people living on this small island. It's 30 kilometers north-south and 20 kilometers east-west. Yeah. So as I told you, Mayotte is a French department. So it's part of Europe. We have to apply the WFD, and our team was in charge of developing its monitoring system. So we had to analyze the water quality of these streams. This is the monitoring network that we set up quite quickly. And what you can see, the different colors, in fact, are in purple, it's the official one, but we wanted also to find reference conditions here in green, and also very polluted areas, just to have a good gradient of all the conditions. So we went there, we sampled, we sequenced all that, and we calculated an index, which is the IPS, so the same index that you're using in Serbia. When it's 1, it means that it's a very bad water quality, and when you are at 20, it means it's a very good water quality. On the horizontal axis is the index that you produce with microscopy, and in the vertical axis, it's the molecular index that we produce with DNA. And you can see that there is a pretty nice correlation between both methodologies. You can see here the R square. Good correlation, but what we could see is that the dominant taxa between both floristic lists were different. Each time when we looked at the molecular floristic list, we often had big species. These were the dominant species with DNA, big species. Whereas it was the contrary, we had mostly small species with microscopic countings. And so we had this, so yeah, maybe I can stop here, or if you want, if that was a plan. No, because I have it just here, but it doesn't matter. Do you have questions about this first part? No? As you want. But just to introduce you that we had this hypothesis that there was an overexpression of the large species in our sequencing data compared to what we have in microscopy. Questions about that? As a result, so we can continue if you want. I need, I think, yes, 10 minutes after. Do I have 10 minutes? 10 minutes? So, okay, let's go
yeah.
for coffee.
Let's go for
Okay,
coffee.
perfect.
Then we will continue.
Okay, that's fine.
Nie.
Okay, I think everything is here. Everyone is here, sorry. So let's have a look to this last part, the impact of biovolumes on the floristic lists. So this is a paper of Valentin Vasselon, and I repeat the questions. The hypothesis is that when you have big diatoms, like this big Pinularia viridiformis, so you can see here, this one is exploded, the fructule is exploded, and the chloroplast is splashed away of the cell. And you can see that the chloroplast is very big. And the question is, do we have genome copies, and do we have numerous genome copies for a big cell compared to this small Acanthidium minutissimum? Is there a relation between the biovolume of the cell and the genome copies? And so so this is a diatom, you open the diatom. In this case, you have 2 chloroplasts. And so we can imagine that in one chloroplast, you have a certain number of genomes. And so we have seen yesterday that in a genome, we have a single copy of RBCL. So we should have this equation with the number of chloroplasts multiplied by the number of genomes multiplied by the number of RBCL copies in a genome. But here, this number is 1. But the question is here, the number of genomes per chloroplast, and is there a relation with the biovolume of the cell? So what did Valentin Vasselon during his PhD? He took 8 different diatom cultures of different species, and these different species had different size, different biovolumes. They were cultivated in triplicates, but what you can see here, you have 3 small triplicates of cultures, and he looked day after day how the number of cells per culture. So you can see here that the culture is growing. There is an exponential phase, and then it should have a plateau and then decrease. And what he did for each day, he took, for each triplicate, he took a small sample to make a DNA extraction, to make a qPCR, to estimate the number of copies, to estimate the number of, of copies he had per cell. and in the same time, he took a small part of the culture and looked at the number of cells per milliliter. And this has been evaluated at 7 different dates that you can see here. So 1, 2, 3, 4, 5, 6, 7, yes, 7 dates. He did this extraction and legal fixation to look at the number of cells and look at the number of copies per milliliter in order to have an estimation of number of RBCL copies per diatom cell. Okay, here is the results. What you can see here on this horizontal axis, you have the biovolume of the cell. So you can see here on the small biovolumes, you have, for instance, these small species, Nichia conspicua, Acanthidium minutissimum, and on the right, you have big cells, big species like Pinularia viridiformis and Ulnaria ulna. And this is a logarithmic scale. And on the vertical axis, you have the number of copies of RBCL the number of copies of RBCL per cell. And you can see that there is a nice correlation between the cell biovolume and the number of copies per cell. And it's a log correlation. So based on this correlation, we can make a correction factor to transform DNA inventories to make them more similar to what we can have in to microscope inventories. And this enables to calculate diatom indices based on these transformed DNA inventories. So if you look at Diatom, each species, in Diatom you have for each species the biovolume, but also a correction factor which is calculated based on this on the formula of the regression you have between these two in these two axes. Sorry, it's here. So for instance, Acanthidium minutissimum, if you want to transform into DNA, into microscopic-like data, the correction factor is small. It's not the case of Pinularia crossfire, which is a big diatom. If you want to make it more similar in terms of diatom floristic list, like microscopy, you have to divide the number of copies or the number of reads you find in DNA with a much bigger correction factor. I'm going to give you an example, just to be sure you understand. Let's transform a DNA inventory into a microscopy-like inventory. So let's imagine we have a sample with 4 taxa, Acanthidium minutissimum, Amphora pediculus, Navicula cryptotenella, and Melosira varians. Here, this is the inventory that you have with microscopy. And here, this is the inventory you have with DNA. So you can see here that the dominant species in microscopy is Acanthidium minutissimum. And then we have a smaller percentage for Amphora pediculus and Navicula cryptotenella. When you look at the inventory just after the bioinformatic pipeline without any transformation, the inventory is very different. The dominant species is Melosira varians, which is a big species. You can see here the biovolume of Melosira varians is 14,000 micrometers, cubic micrometers, whereas Acanthidium minutissimum is only 76. So what you have to do is to look for the correction factor in Diatom for each species. So you see small correction factor for Acanthidium minutissimum and Amphora pediculus, which are small species, and big correction factor for Melosira varians, which is a big species. And so you simply divide the percentage of sequences of Acanthidium minutissimum by this correction factor, and you have this transformation, this transformed floristic list, which looks like the microscopic one. You can see here, we have 70%, 10%, and 18%, and you can see the 8%, sorry, 7%, and the gap between DNA and microscopy is also reduced. So this was for Mayotte. But what about Balkan region? So I'm very keen to wait for the end of a BioLawet project, because I'm sure we will be able to do something similar and probably even better. But I'm sure, sorry, I'm sure it will be better. Sorry. Do you have questions about this part? No? Sorry, here. Maybe go on.
Okay. Okay. For how many species did you calculate the correction factor?
So in fact,
For all species in reference library, or?
probably not all, but almost all. In fact, each time I integrate a new species, I look for the biovolume. And so it's easier after, if you have a biovolume of a species, you get the correction factor with this simple correlation. The formula is here, in fact, the formula for the calculation of the correction factor.
One more
for
question, please.
freshwater rivers, it's not. I don't think it's a big. I think it's working quite well. If you have a biovolume, easily you can get correction factors. It's not a limiting thing. Yeah. So John.
We have two questions from the Zoom. How you calculate volume of diatoms?
It's based on literature. So we open the books, or we look at the publication if it's a new species, and in the description of the species we have this data, so we can calculate the biovolume, and it's based on geometrical shapes. So people who do phytoplankton do know that. Andreas, you know how to do that.
One more question. How this correction factor for the Mayotte Island reflect on the index calculation, the ecological status assessment?
Ecological status assessment. Where is it? Here. So in fact, here we use the IPS. IPS is a Diatom index which has been developed for mainland France. It was in the beginning of our work. We wanted to go quite fast and to see if we're giving some preliminary results. We developed after that, so it's working quite well. IPS is a metropolitan, so it was developed for European rivers. We use it in tropical rivers. It's not the best adapted. We had a lot of work, especially of Kalman. He's here. He developed a Mayotte index, so really adapted to the rivers of Mayotte. And maybe Kalman, you can answer that. This nice question about the development of Diatom indices in Mayotte. Sorry, are you ready for answering this question?
Yeah. Can you repeat the question?
Oh, sorry. No, sorry. No, but
reflected on the index calculation.
no, but maybe the strategy that you used to develop an index based on for diatoms for Mayotte Island, and it's very particular rivers and with special species, a special gradient.
Yeah. So
Shortly.
yeah, basically we use two methods. One is a classical one where we define the ecological optima and the tolerance of the species, and we define an index only yeah, for Mayotte. And we also had, but this is not related maybe to this topic, but we use traits to we define the traits of diatoms, and we did like this two kind of indices. But yeah, because I also wanted to ask something that maybe this can trigger some discussion. because here you show this correction factor. And maybe those who also count phytoplankton, they know that when we count phytoplankton, we use the biovolume. And at the end, we calculate a biomass or biovolume. So when we count diatoms, we have only relative abundances. So why don't we use a correction factor for the morphological results to correct it, to make it more similar to the biovolume? So for example, from an ecological point of view or a perspective, maybe it has more sense. I don't know. What do you think about that?
I totally agree with that. I totally agree. Yelena, you can say, and Daniela also. With diatoms, we count frustules, whatever they are small, like Acanthidium minutissimum, or big, like Melosira varians. And that's the tradition, but probably tradition needs to be modified. And probably phytoplankton, where we use these biovolumes, are much more clever. And yeah, I agree with your point of view, Kelman. And so maybe yeah, the DNA data are more okay.
May I give a comment?
Yeah.
I think the Kalman is totally right. When we count phytoplankton, we need to measure at least 20 or 30 specimens of each species to be able to calculate biovolume. Or after sometimes when we have a good database from these sampling sites, maybe we can use the existing values not to measure each time. But for diatoms, which can be from very large to very small in the same population, depending on the mitosis, it has more sense to use biovolume, but it won't be so easy for routine biomonitoring.
Exactly.
So that's probably we have the resistance from that side to suggest this method.
Yeah, good points, Milos. Yeah. Do you have other questions?